So we're going to start I keep saying chapter. We're going to start lecture 7 here. This is the first of 3 chunks for lecture 7. This first chunk is actually going to be quite brief and just set up the general premise and background for this material. The second 2 chunks will be a bit longer and get into some of the meat of the material we're going to cover here. This is the second half of chapter 7, incidentally, and we're going to continue in general to talk about different mechanisms for controlling and regulating enzymes. So overall, across all 3 chunks, we'll begin talking about the regulation of enzymatic activity in very broad terms. We'll give some definitions of functional terms, and we'll focus primarily on phosphorylation and zymogens. In the second chunk, we'll talk about active sites and what goes on in the active site, and we're going to use a classic enzyme, chymotrypsin, a digestive enzyme, as a specific example for an enzymatic mechanism where we will look at the actual biochemistry that's occurring between the substrate and the side chains of the amino acids of this enzyme right there in the active site. We'll end this lecture series talking about different types of catalyzed reactions and coenzymes. Enzymes cannot always do it on their own. Sometimes enzymes require help to perform catalysis, and that help largely comes in the form of small biomolecules called coenzymes. So last week, and as we watched our lectures online, we discussed the regulation of enzymes from the allosteric enzyme perspective. Specifically, we talked about the concerted and sequential models as 2 different ways by which we can achieve positive cooperativity and regulation, inhibition and activation of allosteric enzymes. Today, we'll start off with talking about some more dramatic regulation, essentially how we can turn enzymes on and off. Period. Turn an enzyme completely on and completely off. None of this, it starts off kind of hard and gets easier stuff. We're talking about a true toggle switch between 2 states, and that will be phosphorylation. We'll also talk about a mechanism by which we can synthesize through transcription and translation an inactive enzyme that is completely non-functional, ship it to wherever it's meant to go, put it in the place it's meant to be active, and then pull the pin, essentially, and irreversibly activate this enzyme once it's where we want it to be. A very potent way of regulating enzymes, especially digestive enzymes. This overall mechanism of regulation falls into the class of Zymogens. And then again, we'll talk about how enzymes work in detail. We'll talk about an enzymatic mechanism as a specific example of how enzymes can, 1, promote the formation of a transition state, and 2, thereby catalyze biochemical reactions. At this point, hopefully, we're comfortable knowing that how enzymes work in general is by lowering the energy of activation, that delta G double dagger. Enzymes make transition state and thereby create reactions that will occur more rapidly and more easily. We know that enzymes recycle, that when an enzyme is done doing its job, it's no worse off or not in any way modified compared to how it was before that enzyme did that job. And really what we're starting to get at here in these lectures is how does all that happen? What is the mechanism by which an enzyme can lower the energy of activation? How is it that an enzyme can help make transition state? And why do enzymes recycle? How come nothing happens to them? How is it that they can regenerate to the point at which they started? So we'll answer those how questions of all this material in these 3 lecture chunks. So let's talk first about phosphorylation as a means by which we can regulate enzymatic activity. Phosphorylation is one of the most common, one of the most potent, and one of the most reversible ways by which we can regulate an enzyme. And I always like to think of phosphorylation as a light switch. Just as light switches allow us to turn lights on and off, and we can toggle between the 2 as often as we'd like, phosphorylation offers the same level of control. We can toggle enzymes on and off, and we can do so reversibly as often as needed. The amino acids specifically which can be phosphorylated are serine, threonine, and tyrosine. So we can look at each of those examples here. Here's the serine side chain. It's a very simple side chain, a CH2 group followed by a hydroxy group. The rest of the protein is kind of up here, the chain of amino acids, and this is the single serine side chain. And we can remove that proton and add a phosphate group, an inorganic phosphate in its place, and this is phosphorylated serine. We have a hydroxyl group on threonine as well, and the same idea. We can remove that proton and pop an inorganic phosphate in its place. And starting to see a theme here. Tyrosine also has a hydroxyl group at the end of that aromatic ring, and we can remove that proton and put an inorganic phosphate in its place. So these are the 3 amino acids that are commonly and easily phosphorylated. The source of that phosphate group can almost always be ATP. Sometimes inorganic phosphate itself is harvested from the environment and tacked on, but most of these reactions require energy, and so by using ATP as the phosphate donor, we get a phosphate and the energy when we break ATP in half. Key terms here: kinases are a class of enzymes that add phosphoryl groups. So any time a protein has been phosphorylated, the enzyme responsible for that phosphorylation is a kinase. And we talked about the reversibility of this process. What reverses that phosphorylation and removes those phosphate groups are classes of enzymes called phosphatases. So kinases add phosphate groups and achieve phosphorylation. Phosphatases remove phosphate groups and achieve dephosphorylation. The effect of that phosphorylation can also differ from protein to protein. Sometimes phosphorylation turns the enzyme off, and you need to dephosphorylate it with a phosphatase to turn it back on. Sometimes phosphorylation turns the enzyme on, and if you want to turn that enzyme off again, you dephosphorylate it. So different enzymes respond to phosphorylation in different ways, but what's important is that we get this toggling switch effect. Through phosphorylation, many enzymes can be turned on or off through the action of kinases and phosphatases. The phosphorylated state will equal one state, on or off, whereas the dephosphorylated state will be the opposite state, off or on. And that's essentially all we need to know in general about phosphorylation as a means by which we can regulate enzymes. So now moving on to zymogens, this irreversible regulation that allows us to create inactive proteins and then ship them to where they should go before we activate them. If phosphorylation's hallmark feature is reversibility, then zymogens are the exact opposite of that. This kind of regulation is completely irreversible. A zymogen, as a term, refers to an inactive form of an enzyme. So when an enzyme is in its zymogen form, it cannot do anything. It's converted into its active form by some cleavage or cleavages of covalent bonds. So we have to cut the protein to turn it on. The reason for this form of regulation is that quite often, when you make an enzyme, you don't want it to start doing its job right away. Some enzymes are very destructive. This is good for cleaning up tissue damage. This is good for digestion. But making a destructive enzyme right there in the middle of the cell could be very bad for the cell. So oftentimes the cell wants to make an enzyme that it needs, but it wants it made in an inactive form. And then you can go and send it where it needs to be, like the small intestine, and activate it there where it's meant to do its job. So chymotrypsin is an example of an enzyme like this. It is a digestive enzyme. When it is made, it's made in its inactive zymogen form, and we call the protein in that form chymotrypsinogen. That trypsinogen, the -agen at the end, like zymogen, signifies that it is in its inactive zymogen form. In this form, chymotrypsinogen is a 245 residue protein. It's made up of 245 amino acids, and it contains 5 disulfide bridges. So we can represent chymotrypsinogen linearly as this here. We see that it's 245 amino acids in length, and these represent the disulfide bridges if and when the protein is folded into its three-dimensional shape. When chymotrypsinogen, completely inactive, makes it to the small intestine, it's quickly digested, that means cleaved, cut, and trypsin is the enzyme that does that cutting. Trypsin is a digestive enzyme, and it's normally found in the small intestine for digestion. So trypsin cuts chymotrypsinogen. And it cuts it in only one place. It cuts between amino acid 15 and 16. So the action of trypsin creates this version of our protein. We call this protein now pi chymotrypsin. It's no longer chymotrypsinogen because we're going to see in a moment it has some activity, but it's not the complete form of chymotrypsin either. So pi chymotrypsin is active. It contains that first 15 residues. They're just held in place by disulfide bridges. But pi chymotrypsin can't digest any protein. It can only cut one protein. And interestingly, the only protein that pi chymotrypsin can cut is other pi chymotrypsins. So these proteins work on one another, and when they do so, they release 2 dipeptides. They release amino acid 14 and 15 as a dipeptide pair, and they release amino acid 147 and 148 as a dipeptide pair as well. This cleavage of one pi chymotrypsin from another, removing these 4 amino acids, creates the complete, final, and fully active chymotrypsin. And this chymotrypsin can cleave and digest almost any protein found in the small intestine as a method towards digestion of those proteins. So we kind of circle back to the idea here of structure equals function. The initial Zymogen, chymotrypsinogen, had a shape that was completely inactive. The shape of chymotrypsinogen didn't allow it to perform any catalysis. After cut by trypsin, the new shape of pi chymotrypsin allowed it to have activity, but the only thing that would fit in its active site is another pi chymotrypsin. And therefore, pi chymotrypsin could only cleave and digest others just like it. Once digested to that final chymotrypsin state, that was a fully functional enzyme with a three-dimensional shape that allowed full-range activity that could go off and digest proteins as part of our normal digestion process. So this is, again, a very potent form of regulation. You go from completely off, and there's not much you can do about it, to completely on, and there's not much you can do about it either, allowing you to make dangerous enzymes, but make them in a safe way so that later on you can activate them when they should be active. So that's all we're going to cover in this first chunk here. We covered the ideas of phosphorylation and Zymogens as two different general regulatory mechanisms for enzymes, and they couldn't be any different from one another, right? Phosphorylation is a toggle switch, on, off, on, off, on, off, phosphorylation, dephosphorylation, kinase, phosphatase. You could do that all day long and go between the two states as much as you need to. Zymogens, completely the opposite. You're made off, you're turned on, end of story. We're going to move on to talk about coenzymes in just a little bit, and also those enzymatic mechanisms as well, but all of that will be saved for the next two lecture chunks of this series. As always, thanks for watching.